Okay, well, good morning, everyone, and welcome to today's edition of the seminar series. So, once more, our host of lovely links for you to distribute widely. Um, as always, just a quick reminder that we have our workshop on these post-Bayesian topics happening in mid-May. If you are interested in coming, please register. We are also really looking forward to receiving your submission. Thank you to everyone who's already submitted something. We've got some really exciting proposals. Um, as last week, please use the Q&A function to ask talks, and we'll see if we can get to them as they come. And then after the talk, we'll do the usual discussion series. So, over the last few weeks, we've seen more of the theoretical underpinnings of these post-Bayesian objects from a kind of inference perspective. And this week we have Chris Oates from Newcastle who's going to talk to us a bit more about the predictive view of these objects. Chris has been working on the computation of these objects and much more for the past, well, for a while now. And I had the pleasure of seeing him be awarded his Guy Medal in September. So he's been doing some incredible work on this. And so I'm very excited for today's talk. Maybe I'll hand it over to you, Chris, straight away.
Thank you, Jan, and thank you to all the organizers. I'm very happy to be presenting this morning in the UK, but whatever time it is where you are, thank you very much for joining. So um, we've had some really excellent talks so far in this webinar, and I'm really pleased that this webinar series is happening. Just to sort of reveal a little glimpse behind the curtain, I'm trying to persuade these guys that they should set up a section of ISBA for post-Bayesian methods, because there's a huge amount of interest, and I think it'd be really useful for the community. But that's my project to make that happen. So just for today, we're going to carry on with the webinar. And my starting point is generalized Bayes, but we've learned a lot about generalized Bayes over the last few talks. And I'd like to take an epsilon step beyond generalized Bayes and think about what the trouble with generalized Bayes might be, focusing on a case study where generalized Bayes doesn't seem to provide a satisfactory solution, and think about what methods might help us to perform some sort of satisfactory inference in that setting. So I'm trying to avoid doing the obvious thing, which is to have a slide that has the famous George Box quote and then to patronize you, because you all know that the models are wrong. But let me just briefly dwell on why we're in this mess in the first place. And it's kind of like the sociology of how research and science and industry works, right? So you've got researchers that are siloed into different disciplines. And for different disciplines, there are different things that are important, right? So in particular, when people say model, this is one of these overloaded terms that means different things to different communities. If I was, let's say, in the humanities, then a model might be something like some of these pictures on the screen, right? It might be a Venn diagram of some kind with some words in it. And that might be the right way to communicate the kind of ideas and concepts that are important to researchers in the humanities. Or if I was an engineer, maybe a model is a set of physical equations that describe how different forces propagate through mechanical structures that I'd like to build one day. Or maybe if I was a linguist, a model is about how different grammatical structures and so on relate to each other. And my model would be sort of graphically represented like a flow chart. Or I guess I should stop at some point, but if I was you know well, let me stop because you get the idea, right? So what really is the root cause of model misspecification is this overloading of the word model. And we're very typically in the following situation where we're provided with some sort of mathematical description of something, but it's not fulfilling the requirements of a bona fide statistical model. And we have to take that and turn it into a statistical model somehow for the purposes of doing either inference or prediction. So for the purpose of this talk, I'm going to focus on quote models that are deterministic parametric functions that take some sort of input and return some sort of output. Okay. And so those engineering type examples on the previous slide are a good sort of idea to have in mind. You've got something which tells you how a response is supposed to behave in response to an input. And there are some degrees of freedom or parameters that I'll generically denote theta that govern that response. And of course, you're given one of these models and asked to infer the parameters theta and perform prediction, but it's not a statistical model. So that's where some work is required. And generically speaking, we're interested not just in inference prediction, but also in causal inference and causal prediction, right? So in engineering, again, these models are very typically causal models, right? They explain how the laws of physics come together to determine what happens to a mechanical object on application of a force. And in particular, if you changed some aspect of the system, right? So if you removed an important beam from a structure, that would change how the forces propagate, and you could reason about the causal effect of that intervention through the mechanistic model. So these models typically often have a causal content as well. So if we're given one of these, let's say, causal deterministic models, how do we turn that into a statistical model? And the usual solution, which many times is a reasonable thing to do, is to interpret the deterministic part as the mean of a regression model. So let's let p theta denote now a statistical model, which I obtain by taking my deterministic model as a mean and then imagining some sort of error process. So the typical thing to do is to imagine independent normally distributed errors, and that gives rise to a statistical formulation relating observed data to parameters of the mechanistic model. Okay. And we know that this can lead to various forms of misspecification, and a whole community of generalized and post-Bayesian researchers is aimed at tackling these kinds of misspecification problems. I want to focus on a case study that helps to illustrate some of the things that can go wrong, even with generalized Bayes in this setting. This is a case study that I'm sort of fond with because it was part of my PhD research. This is protein signaling. And the systems biology community have their own take on what a model is. And for them, a model looks something like this picture here. Okay. So let me just take a couple of minutes to explain what's going on in this picture. Each shape represents some kind of protein, and the arrows represent chemical reactions involving those proteins, right? So each of these proteins has the capacity to influence the chemical functional behavior of some other protein, and that's indicated by these arrows. So just to take a specific arrow and explain what this means. Here we've got a protein called MEK, and MEK can be chemically altered by the addition of phosphor molecules. They're represented by these yellow circles here. And when MEK is phosphorylated, that changes the conformation of the molecule, revealing some more sort of active parts of the molecule that can then go on to do other things. So the conversion of MEK from the unphosphorylated form to the phosphorylated form is catalyzed by an enzyme, and that enzyme is RAF1 in this case, right? So this arrow here means that RAF1 is an enzyme that speeds up the conversion of unphosphorylated MEK to phosphorylated MEK. Okay. So that's the context. And these models, right, they're not just toy pictures that we can kind of, sneer at. These are representing hours and hours and hours and millions of pounds of people's time and money, because each arrow corresponds to someone's PhD, right? They have to get these molecules, they have to extract them in isolation, they have to examine the crystal structure, they have to check that these things really do bind to each other. A huge academic effort goes into just establishing that there are arrows or not in this picture. So this conveys an awful lot of information, and it's causal information. And that causal information has an important medical use, right? So this red thing here, this represents a drug treatment. So it turns out this protein signaling pathway is a causal driver of various different cancers, particularly certain types of breast cancer. So this ERK, when it's in its phosphorylated state, it can translocate from the cytoplasm, which is where all of this stuff is going on, into the nucleus. And once it's in the nucleus, it starts affecting gene expression levels, which in turn causes global changes in what the cell's doing, typically leading it to divide more than it should. That's the root cause of a cancer in this case. So, so drug developers had the idea that if they can design a small molecule called a MEK inhibitor, which binds to phosphorylated MEK and stops its catalytic activity in terms of phosphorylating this ERK protein, then that might actually be a useful targeted therapy for these particular cancer subtypes. And indeed, this works in the real world. People are given MEK inhibitors, and models like this are used to understand and reason about the effect of giving a MEK inhibitor to a patient. So to make a more quantitative statement, we obviously need to turn a picture like this into some kind of mathematical equation. And that can be done reasonably routinely. So if you're familiar with mass action chemistry, whenever there's a chemical reaction, you can write down mass action chemical kinetic equations, associated rate constants, and try and infer those rate constants. You get a lot of equations that way, and things can be simplified a little bit in the case of enzymatic kinetics, because essentially some reactions are faster than others, and you can do a sort of fast, slow approximation that reduces the number of rate constants to be inferred. So broadly speaking, I can take this picture on the left and translate it into a system of 11 different variables that evolve in a deterministic way through time, parameterized by also 11 rate constants, or sort of combinations of rate constants. Now, just for the purpose of showing you something in this talk, I haven't got real cell data that we're going to try and fit to this model, because that's a really messy problem, right? What we're going to do is we're going to simulate from something, and we're going to try and learn these parameters theta. And this is going to be illustrative of the kind of problems that you face in the real world, in particular in systems biology. So as a thought experiment, let's generate data. Let's generate protein concentrations through time, and we'll try and infer the parameters theta from those data. And what I'm going to do is I'm going to introduce a tiny amount of model misspecification, right? So it's going to be a tiny amount. I'm not going to change which proteins interact with which other proteins. I'm not going to introduce any latent confounding or anything. So it's really just this isolated system. It's highly idealized. But what I'm going to do is, instead of simulating concentrations U from an ODE, I'm going to simulate from an SDE. So in other words, I'm going to add on a small amount of diffusion to this ODE and turn it into an SDE. But it's going to be an incrementally small amount of diffusion. But I'm going to use the ODE plus Gaussian measurement noise to fit the data. And the data come from an SDE plus Gaussian measurement noise. So the measurement error model is the same. It's always Gaussian noise on the protein concentrations. The only difference is whether there's intrinsic stochasticity or not in the evolution of the protein concentrations. So this is what happens. The red crosses here are my data. So these are the protein concentrations plus a small amount of Gaussian measurement noise. The black lines are the evolutions of the protein concentrations. And if we take a look at the first and third rows here, these are the fitted posterior predictive means to those data, right? And so basically the red dashed line and the black curve are indistinguishable. The posterior, is a perfect fit to these data. Everything looks very nice. And underneath, on the second and fourth lines, what I'm showing you there is the predictive mean and the true behavior under the action of one of these MEK inhibitors. So what I've done now is I've taken the ODE system, I've pretended that I've got a MEK inhibitor, I've applied that to the system, and I've simulated from the interventional model. Right? So I can make a causal prediction about what happens under the action of a MEK inhibitor, because I had a causal model in the first place. And my predictive mean, the red dashed line, now looks pretty similar to what happens in reality under the action of the MEK inhibitor. Let's zoom in a little bit, because it's quite hard to see here. So let's take this panel down here. So this panel represents the evolution of the concentration of the phosphorylated MEK-ERK complex under the action of a MEK inhibitor. Let's just zoom in slightly. And you can see that the predictive mean, which is this dashed red line, is in good agreement with the actual evolution of this concentration of phospho-MEK-ERK. But let's zoom in even more. And what you can see is underneath that red dashed line, there's actually a gray shaded region as well. And this gray shaded region, that is the 2 sigma posterior predictive credible set, the pointwise credible set. And actually now you can see everything's gone horribly wrong. Because although the predictive mean is sort of doing generally the right sort of thing, protein concentration increases in some way with time, the posterior credible sets are vastly miscalibrated, meaning that the posterior is effectively certain that the protein concentration will evolve in this way, and it actually doesn't evolve in exactly that way. So, you all know that that can happen when you've got a misspecified model, but to see essentially a very tiny amount of misspecification that was completely undiagnosable when you looked at the fit to data, having such a catastrophic effect on the collapse of the uncertainty, I think that's quite shocking. And that happens here because the predictive distribution is driven by a deterministic mathematical model. If you've got no parameter uncertainty, you've got no predictive uncertainty. And so if your prediction is wrong, you're catastrophically overconfident about something that's wrong. So what can we do about that? Well, obviously generalized Bayes is around, but let's focus on just Bayes to start with, because the usual remedy that Bayesians propose to problems like this is to be more Bayesian, right? So actually, you probably ignored some things that were important. Let's build a bigger model, integrate in more sources of uncertainty, and then for prediction, we'll integrate everything out that we don't care about. That should give us a better calibrated uncertainty. So let's take that idea and explore whether that can solve our problem in this context. So when we're dealing with Bayesian inference for a deterministic parametric model, one of the most common approaches is due to Kennedy and O'Hagan, and it's essentially modeling the model misspecification as a latent variable. So let me just spell out how that works. So my parameters are going to be theta. We're going to assume we've got IID data. We've again got this parametric function, which is a sort of domain-specific model. And the key concept in Kennedy and O'Hagan is to introduce this object here. So this is a mapping from inputs to outputs that we can think of as a residual. It's going to act to soak up any differences between the real world and this model. And of course, being Bayesian, if something is a variable, then you have a distribution on it. In this case, we place a prior distribution on this residual. Being function valued, a nice convenient choice of prior would be something like a Gaussian process. And that leads to this augmented statistical model, which is exactly what we had before, but now we've got this non-parametric function, which serves to mop up any kind of model misspecification that might be present in the first term. Okay. So this Kennedy and O'Hagan approach is widely used when scientists, engineers build these parametric simulators of physical processes, and they're trying to reconcile these models with data. And they're very effective when it comes to predicting real-world observational data. But there are two main drawbacks with this approach, and one of them is really important. So the first drawback is just the practical thing, right? If you've got a high-dimensional latent random variable, like a function-valued random variable, you have to learn that somehow, and typically that requires enough data. So maybe if you're in systems biology or something, the data are actually quite expensive and you wouldn't want to have to estimate a high-dimensional latent random variable. But that's kind of just a soft argument. A really hard argument against this approach is that you can't do any causal prediction with it. So what I mean here is that you have data from an observational regime, so you can learn what this model misspecification is in the observational regime, but you don't typically have any data from the interventional regime. That's the whole point of causal prediction. You want to predict what would happen if you made the intervention. So you've got no way of working out how this part of the model ought to change under an intervention, meaning that you just can't do any kind of causal prediction with Kennedy and O'Hagan. So that's a shame, and I don't know any way to fix that. Let's now jump to generalized Bayes and see what happens with generalized Bayes. So, I don't really need to say too much about generalized Bayes because it's been introduced in a much better way than I'm about to, but just to at least put an equation on the board that we can look at. We've got the parameters, the data, and now the key ingredient that separates generalized Bayes from standard Bayes is this idea of a loss function, a mapping that takes parameters and data and returns a scalar value that quantifies the loss associated with a particular parameter on the basis of the data that you have. It's a sort of pseudo-Bayesian type method, so there's something that we can call a prior, and the generalized posterior is defined as solving a variational problem that balances the average data fit with the distance in some sense to the prior distribution. And just to skim through this, because it's all been covered in much more detail in earlier talks, standard Bayes drops out for the negative log likelihood loss. There's also this choice of regularization strength, and we can choose that to be 1 if we want to recover standard Bayes. But of course, there are lots of other choices of loss functions that have been discussed at length, and they can buy you things like robustness to outliers and so on. The big issue with generalized Bayes, which is the one I want to highlight as being problematic in the motivating systems biology context, is that for most typical choices of loss, and most typical choices of the learning rate slash regularization parameter lambda, you get some sort of posterior concentration, or in other words, a collapse of the generalized posterior to a point mass on some parameter vector. Now, that's something that's been studied theoretically by people like Jeff Miller, for example, but it also is the case that for finite size data sets, you do tend to have a concentration of some kind, vanishing uncertainty as you get enough data. So that's problematic because it means that in a misspecified deterministic modeling context, you're going to be predicting using the model with a single value of the parameter, which is going to generate a line with zero uncertainty around it. Okay, what could solve that problem? Well, maybe we can play with the learning rate. So this is sort of like the really important part of generalized Bayes is choosing this learning rate somehow. And Ryan gave a great talk last week about how to do this, so I won't go into any details. But just to say, one thing is it's sort of open, right? How to do this? There's lots of ideas around. Some of them are somewhat computational, and I think it's fair to say that the story is a little bit complicated when you compare it to standard Bayes and Bayes theorem, which you write down on the blackboard in one line. But let's just leave it there and say it's sort of hard. So, unless you're going to think very hard and very carefully, you're going to have collapsing generalized posterior uncertainty and have predictive overconfidence, right? Because in the cell biology example, for any value of the parameter, the predictive mean under intervention would have been wrong. And if you've got vanishing parameter uncertainty, you've got vanishing predictive uncertainty. So the problem remains even in generalized Bayes. So that led us to think about a different approach that has the flavor of a post-Bayesian method, but is not generalized Bayes. And I'm not actually sure this name is helpful, but it's the name that we're currently using. So I'm going to use it here. We're going to talk about methods that are called prediction-centric. And so let me just define what a prediction-centric approach is for the purposes of this talk. And then maybe we can think about what a better name should be. So we're supposing that we're given a statistical model as a starting point. And, presumably this model is useful in some way. It has some useful sort of causal content built into it, but we suspect it's misspecified. The first thing we're going to do is we're going to lift that statistical model and turn it into a mixture model. Okay. So if you actually had a misspecified model, then lifting it into a mixture model gives you some way of mitigating the effect of the misspecification, right? So just to take a simple example, if I've got some data distribution that's not really very Gaussian, but I've got a Gaussian model, well, the Gaussian model might not fit very well, but a Gaussian mixture model is a universal approximator. So I can definitely fit my data distribution with a Gaussian mixture model. The story is a bit more complicated when you've got, complex models that involve, ODEs and so on. But nevertheless, you get strictly greater flexibility by thinking of mixtures of your original statistical model. And now the key thing here is the mixing distribution Q that says how we should assign weight to different parameter values in the mixture model. And step 2 of this prediction-centric approach will be to learn Q somehow. So what we're going to focus on is matching this distribution, the mixture model, to the data distribution in some way. Okay, so this is a glorified way of fitting a mixture model, but it's a glorified mixture model that actually can involve quite complicated statistical models as the mixture components. Now, this is not a new idea, obviously. It can't possibly be a new idea. And I just want to illustrate some instances where this idea has already appeared in the literature. And if we've got any time left at the end, I'll discuss 1 or 2 things that we did which we thought were useful in this context as well. So the first way that I'd like to mention is known as non-parametric maximum likelihood. So non-parametric maximum likelihood starts with a mixture model of this form, and it selects the mixing distribution Q by minimizing this sum, effectively, of negative log mixture likelihoods. Okay. So you've seen generalized Bayes talks before. This is somehow an empirical approximation to a Kullback-Leibler divergence between a data distribution and the mixture model. So that's sort of a nice thing to do, but it has some challenges as well. And I guess there are 2 main challenges here. One is that you can show the solution of this thing actually is degenerate in the sense that the minimizing Q will be supported on a finite number of points, essentially. And that's computationally challenging because it actually means the optimization to find Q is a sort of horrible non-convex thing where you can use numerical methods, but you typically don't have guarantees that they're going to converge. But the other problem is statistical, right? So you don't have this nice post-Bayesian idea of updating belief distributions. If I start with a prior and I see some data I'd like to have a posterior of some kind, you can't really get that when immediately after seeing some data, you end up with a discretely supported distribution that's not going to be absolutely continuous with respect to anything else. So that's a slight drawback with non-parametric maximum likelihood, but it does fall into this broad umbrella of prediction-centric approaches. So, okay, if that if that hasn't got convexity or absolute continuity, just regularize it. And these ideas have appeared not so much as general statistical methodological proposals, but as particular things people did in the Gaussian process community to make things work. So there's a couple of papers here that I want to highlight that studied effectively just KL regularized versions of non-parametric maximum likelihood. There were some slight sort of extra things going on in these papers. For example, there was some sort of tempering going on the likelihood, but effectively we can think of these as KL regularized versions of non-parametric maximum likelihood. Okay. And the KL regularization overcomes both of those issues that we saw before, right? So there's some sort of convexification going on, meaning that you could, in principle, obtain the minimizer Q using the right techniques. I actually forget whether these papers used the right techniques, but in principle you can. And the Q that you get will be absolutely continuous with respect to Q0, and it would have to be, otherwise the KL term would blow up and you wouldn't be at a minimizer. So that's really nice. But the sort of the gap here is how to do the computational bit, right? And that's where my personal interest starts in this project, because I'm a computational statistician by training, and I like to do things that that are sort of practically motivated. Let me just mention one more paper. So this actually came out very shortly after the paper that I'm going to mention later that we worked on. It's looking at the negative log Bixter likelihood as the predictive fit term with a KL penalty again. But this KL penalty is slightly different to what we've seen before in that it's not regularizing towards the prior in some sense, it's regularizing towards the standard Bayesian posterior, which is an interesting take on things. I think the authors were saying with this that they want some sort of robustness to model misspecification, but they didn't want to depart too much from the standard Bayesian framework. And that motivated regularizing towards the standard Bayesian posterior. They also pointed out that you could use effectively any proper scoring rule for this predictive fit term. It wouldn't have to be negative log likelihood. One further thing that these authors did was to use parametric variational inference for computation. So essentially restricting the set of allowed Q to some parametric distribution class that can be optimized using sort of off-the-shelf techniques. So that's a brief summary of what exists in the literature. What I'm going to do in the next 10 minutes or so is just briefly explain our take on prediction-centric alternatives to generalized Bayes. And we're really going to just try and make two improvements to what I've just described. So the first improvement relates to this kind of approach, regularized on parametric maximum likelihood. We'd like to be able to efficiently compute the solution of these kind of problems. And luckily, there's now a lot of tools available to solve variational problems like this that derive from gradient flows. So we're going to see whether ideas from gradient flows can be brought to bear on solving problems like this. And compared to predictive variational inference, we're going to regularize not with respect to the standard Bayes posterior, but with respect to some sort of prior. And that's going to give us this idea of updating belief distributions in the sense that the solution of these variational problems will be absolutely continuous with respect to some prior. Okay, so this, as I say, is our take on prediction-centric and centric quantification, and our in this case means Jiang, who's a postdoc with me at Newcastle, Jeremias, one of the organizers of this webinar, and Sam Power from Bristol. So the setting again is, for now at least, IID data. If we've got time at the end, I'll explain how you can go beyond IID data, but I think you don't lose too much of the story by focusing on IID data here. We're going to be matching this mixture model to the data somehow, and to do that, we need this idea of the empirical measure of the data set. So this P sub n is just a uniformly weighted collection of point masses at the data distributions. It's effectively a histogram of your data. That's our mixture model from before. And we're going to define what we're going to call a prediction-centric posterior to be the solution of this particular variational problem. So we've got KL regularization towards some reference distribution, which we can call a prior. We've got some sort of learning rate lambda, and we're going to measure predictive fit using something which I'll define in a second, but it's called the maximum mean discrepancy between the empirical distribution of the data and this mixture model. Okay? And we're interested in the mixing distribution Q, for which this objective is minimized. Okay, so just a few remarks to unpack this. MMD is a family of statistical divergences that are a way of measuring the similarity, or I should say difference really, between two different distributions. And I'm particularly fond of these MMDs because they are characterized by an object called a kernel. And it has the property that you can write down algorithms that apply to any kernel, but by choosing the kernel in various different ways, you can control the sort of the strength of the topology that this MMD is inducing on the set of distributions, right? So for different choices of the kernel, MMD can either be a very weak notion of the dissimilarity between distributions, or it can be very strong. And you don't need to rewrite your algorithms. You write the algorithms once and just change the kernel. So for me, this is a really beautiful type of statistical divergence to work with. And it's also going to give us control over the robustness to outliers of our prediction-centric posterior. So it's sort of well known that for particular choices of kernel, if the empirical distribution of the data contains some outliers, then this MMD term is basically going to ignore them. So there's lots of benefits to using MMD in this context. The one I'm going to focus on is the computational benefit, right? So actually, MMD is computationally explicit, and we can get various different functional gradients of it, which are useful for constructing algorithms like gradient flows. I've discussed regularization towards a prior and how that means that the prediction-centric posterior has this interpretation of updating belief distributions because of the absolute continuity. And let me just talk about the learning rate. So I highlighted the learning rate as essentially the big challenge for generalized Bayes. And I think Ryan's talk supported that this is an important problem. The trouble with generalized Bayes is that if the learning rate is too small, the generalized posterior collapses to a point mass. That doesn't happen here, right? So let's think, what happens if the learning rate is too small? Well, then we're just minimizing this MMD term here, okay? Now, let's go back to this toy example of, I've got a Gaussian model, and my data are this sort of non-Gaussian thing. So if the best fit to my data is from a Gaussian, then sure, this thing will collapse to a point mass on a particular mean and variance for a Gaussian that happen to fit the Gaussian distribution of the data. But if the data are not a Gaussian, then I'm not going to get the best fit by using a Gaussian. I'll get the best fit by using a Gaussian mixture model. So the solution of this variational problem will not be a point mass distribution for Q. It will be something richer than that. So I don't have this collapse of uncertainty when the learning rate goes to 0, which makes it conceptually very different from generalized Bayes. It actually works really nicely in the systems biology example that I showed you earlier. So without really doing too much tuning or anything at all, we get these nice predictive distributions with sensible looking point-wise credible sets under the interventional regime. There's a few things that need unpacking here. For one thing, this isn't IID data, so I need to tell you how we applied this method for IID data, but we'll come back to that. The other thing to notice is that I think the predictive mean is maybe slightly less accurate compared to generalized Bayes, but if we're trading off a small amount of accuracy versus meaningful uncertainty, I think that's probably a good position to be in. Okay, zooming in on that panel again that we saw before, this is the Mekirk complex that was sort of whose trend was well predicted by generalized Bayes, but the uncertainty was hugely overconfident. Now we've got a trend that again is sort of increasing in some way, but the uncertainty is much more commensurate with the size of the actual error between the posterior mean and the true protein concentration. So maybe now I'll take 5 minutes or so to unpack what happens under the hood and really focusing on the computation. How do we get out these prediction-centric posteriors? So as I mentioned before, this maximum mean discrepancy is a family of statistical divergences that are characterized by something called a kernel. For those of you that have not seen these before, a kernel is a symmetric positive semi-definite function that takes two data points as input and returns some measure of similarity between the data points. So there are lots of examples of these, but if we just take a look at one example, this would be called the polynomial kernel of degree 2. Right, so for two data points, and I'm assuming these are scalar valued for illustrative purposes, I could look at the product of the data points and then square the product of the data points. It's just a toy example. Now, for every kernel, there's a whole family of functions that I can build by taking these kernels, fixing one argument, and then treating those as my building blocks, right, by varying the first argument and fixing the second argument. So I get these building block functions that I can translate and I can superimpose to generate interesting functions. In this case, the functions I generate would actually just be quadratic polynomials, so it wouldn't be very interesting. But for other choices of kernels, you can get much more interesting function classes. And those function classes are known as reproducing kernel Hilbert spaces. I'm not going to go into any detail at all about this, but the reason why we're introducing reproducing kernel Hilbert spaces is that the set of probability distributions doesn't have a nice vector space structure, right? And if we want to start doing algorithms that are computationally nice algorithms, then we need some sort of basic mathematical structures like vector space structures. So the idea of MMD is, although our distributions themselves don't, live in a vector space, we can take them and put them into a vector space. And in particular, we can put them into not just a vector space, but a normed vector space, and even a complete normed vector space within an inner product. It could be a Hilbert space. It could have all of the mathematical structure that we would ever want to use computationally. And that's exactly what happens with MMD. So I can do that in one step, or maybe two steps, by defining this mapping here, which takes a probability distribution P and maps it into a Hilbert space of functions via this map here. This is called the kernel mean embedding. So what I do is I just integrate out one argument of the kernel with respect to the distribution that I'd like to embed. And now this object here, which I'm not going to sort of define technically at all, is a function, right? So I take some input, I plug it in there, and I get a number out. So this is a function now. And because I've now got a functional representation of my probability distribution, it's much easier to do things like take norms, measure distances, take angles, and so on, because I'm in a function space that has all of that mathematical structure built into it. So we define the MMD between two distributions, P star and P, as just the distance as measured by the norm between the mean embeddings of the two distributions. So in that sense, it's quite straightforward, but this also translates into computationally explicit algorithms, right? So for this kernel, for example, the quadratic kernel, the maximum mean discrepancy ends up being exactly just the norm difference between two vectors. And what's in these vectors? It's just the first and second central moments of the distributions. So we're just saying two distributions are similar if they've got similar first and second central moments, which is a very intuitive thing to be able to describe. But of course, the power of MMD is that we can change the kernel. Pretty much all of the maths stays the same, but we can change the topologies that this MMD actually induces on the set of probability distributions. So the quadratic kernel tells us about two moments, but it doesn't actually separate different distributions, right? Because we can have two distributions that have identical first and second moments, but differ in the kurtosis or something. And this MMD wouldn't be able to tell us that those distributions were different. So what we would like is to choose our kernel in such a way that the MMD is zero if and only if the two distributions happen to be exactly the same distributions. Kernels that have that property are known as characteristic kernels. And luckily, there are a whole list of very mathematically simple characteristic kernels. Squared exponential or Gaussian kernels, for example, have this property. So we can do that, and we can get nice topologies whose strength can even be varied sort of more subtly depending on exactly how the kernel is chosen. Okay, so now we know what MMD is. how do we compute the MMD when we're working with this mixture model that we have in the prediction-centric approach? So the MMD itself is an integral because of the mean embedding, and the mixture model itself is an integral because we're integrating over the mixing distribution, Q. So there's a double integral now appearing in the kernel mean embedding. And I can think about this actually as the mean embedding of the mixture model is a mixture of the mean embedding of the model. Okay, so I've just interchanged some words, which corresponds to the interchange of integrals. And what that means is that when we want to evaluate the MMD between the data distribution, which I'm going to denote by P star, and this mixture model, we can do everything very explicitly. So the squared MMD is the squared norm between the mixture models of the mean embeddings. And if we do a bit more maths, we can actually recognize that as just a quadratic form in the mixing distribution Q for a particular integral kernel that I've denoted kappa sub P star. For anyone that has sort of interest in computational stats / machine learning, you might recognize this object as an example of a Stein kernel. That interpretation isn't really necessary to understand what we're doing here, but it's just nice to see so-called kernel Stein discrepancies emerging in a seemingly unrelated context here. But the important observation is that this MMD is a quadratic form in Q, albeit quadratic, over a particular set of probability distributions. But glossing over the technicalities, if we've got a quadratic, then minimizing quadratics is a nice computational problem, right? Because these are convex objectives, and if we just do gradient descent, we'll eventually get to a minimum. Let's just also say that, assuming that we have chosen a characteristic kernel for the MMD, then this quantity here will be, So assuming we've got a characteristic kernel, and assuming that the model is in fact well specified, meaning that there is a particular parameter vector for which the data distribution coincides with the model, then this MMD will be 0 if and only if Q is a Dirac distribution on that correct parameter value. So in the well-specified case, this would actually have the right property of being minimized by Dirac on the right parameter value. Now, of course, we don't have access to the data generating distribution. We just got access to samples from it. So practically, to get the prediction-centric posterior, we'll be plugging in the empirical distribution here in place of the true data generating distribution. So in practice, you can't recover the exact parameter value exactly, but asymptotically, in some sense, everything works as it ought to in the well-specified case. Okay, so if you recall, the prediction-centric posterior that we defined was the weighted combination of the MMD term and then a KL regularizer. And it's that KL regularizer that actually convexifies the problem over the space of measures. So we've got the convex MMD and then the convex regularizer. And that's enough to justify formulating a entropy regularized gradient flow whose solution is the prediction-centric posterior. Now, I think because of time, I'm going to gloss over exactly the details of how to do this, but suffice to say there's a very active subfield of computational statistics and machine learning at the moment that's investigating practical and theoretical aspects of gradient flows. And the prediction-centric posterior is defined by a variational problem that is very well suited to a family of gradient flows called Wasserstein gradient flows, which can be written down mathematically as solutions of SDEs due to a dual characterization between a gradient flow and a stochastic process. And the stochastic process can then be discretized for simulation as you would discretize any stochastic process for simulation. So what we end up with is essentially an interacting particle system that we evolve forward in time, whose solution converges in some sense to the prediction-centric posterior, the solution of the variational problem. And in pictures, this is what that means. So what we're going to do is we're going to initialize some particles somewhere at time 0. And we're going to run some interacting particle system forward in time. And the location of these particles will eventually settle down. And the resulting locations collectively form a discrete approximation to the prediction-centric posterior. If we only use 5 particles, then it's not a particularly fine-grained approximation, but we can use 15 particles to get more resolution and so on. As we increase the particle number, we get a better approximation of the prediction-centric posterior. So we've got a numerical or computational handle on this minimizer of our variational problem, which means that we can access the prediction-centric posterior computationally. Okay. And then just finally, the motivating systems biology example was for non-IID data, right? So when you're in a regression setting, you don't have independence because you've got this x-axis. But you do have conditional independence. So if I told you the value of x, then the responses would be conditionally independent given x. And what that means is that the data distribution, rather than being just some distribution p star, is now going to be a family of conditional distributions that are indexed by the covariate. And correspondingly, when we have a model, that's going to correspond to a family of distributions that are indexed by covariates as well. And we'd like to match this family to the data generating family of conditional distributions. So that, in principle, can make the problem much harder. What we've done is to take what I think in the one hand is a simple / naive strategy to map the problem onto an IID data problem. But actually, I think the strategy has some serious theoretical justifications as well. I think it's probably an idea that can be found, long ago in the literature. But the reference that I'm most familiar with is this recent one by Pierre Alquier and Matthew Gerber. So the idea is that we'd like to have IID data, and we can do that if we pretend that these pairs of covariates and responses are IID draws from some joint distribution over x's and y's. But how do we do that? Because the problem doesn't naturally come with a distribution on the x's, right? In systems biology, you might just measure protein concentrations at fixed time intervals, for example. Well, that's not sampling times from some distribution. It's just measuring times at fixed intervals. So the solution that Alquier and Gerber proposed is that you can use the plug-in empirical distribution of the covariates as a surrogate for the marginal distribution of the covariate in this joint model. And that can be justified from a variety of different lenses theoretically, but practically it's more than sufficient to map a regression problem onto an IID data problem where we can just apply the methods I've just described. Okay. And we do the same thing for the model, right? So we just replace the sort of marginal distribution for x with the empirical distribution of the covariates that we happen to have in our data set for x. Now, you could argue that that's a sort of a natural thing to do. But we've got an extra sort of trick up our sleeves here because we've got a choice of kernel. And sometimes the choice of kernel is a problem, right? You've got to pick a kernel and you've got to commit to something. But in this case, it's a blessing because it gives us some flexibility to actually mitigate the plug-in approximation that we've done here. So when we lift a regression problem to this IID setting, we need to define a kernel on the joint space of covariates and responses. One sort of straightforward way to do that would be to use something like a Gaussian kernel. So here I'm measuring the distance between the covariates and the distance between the responses, and the contributions of these distances depends on a certain length scale parameter, sort of characteristic length scale, separately for the covariates and for the responses. And what we can do here is we can take the length scale associated with the covariates to 0. And what this means is that whenever two covariates are distinct, we basically don't see any dependence or similarity between distinct covariates entering into the MMD. So in other words, we can engineer the MMD to be somewhat insensitive to this plug-in approximation that we're making here when we go from the regression setting to this augmented IID setting. So let me now just summarize briefly what I've said, and I hope some of it was coherent, but I understand that probably not all of it was. I think the main take-home messages here are that for me, I see communities that have an effective and useful definition of model that works for them, right? It communicates the basic information that they need to share with each other when they talk about a model. And then the challenge occurs when we map the domain-specific definition of a model onto a statistical model. So for me, when we develop post-Bayesian methods, we should be targeting specific communities where there's an original definition of model that needs to be made into a statistical model, because the methodology should somehow acknowledge the important information content that's specific to those communities when they build their models. we've discussed these prediction-centric approaches, and again, I don't know if that's the best name. They're certainly not new approaches, but we think they're interesting. They do seem to work in this systems biology example, and they've got some really nice computational properties that make for attractive methodology, in my opinion. Now, there's lots of things that I didn't discuss, and I've given this talk a few times now, and there's always new things that I learn from the audience after the talks, right? So I gave this talk yesterday, actually, at King's College in London, and I learned about Bayesian exponentially tilted empirical likelihood. So this is, another thing that I need to go away and think about. But some other notable omissions from this discussion are things like martingale posteriors, which I know that this webinar will be focusing on in detail in the next chapter. I also wonder whether the huge interest in conformal prediction that's currently going on in statistics and machine learning could have something to say in this context. Lots of interesting questions that we just didn't touch on at all. So if you'd like to find out more about what we did specifically for prediction-centric and scientific quantification, there is a paper. And if you're going along to AI Stats, you'll also be able to see the poster as well. So thank you very much, everyone, for your attention.
Terrific. Thank you so much, Chris. So we'll now take some questions for the remaining 7 minutes. If anybody wants to just unmute themselves to start talking, I think that should be okay. Or maybe raising your hand for some civility. Edwin, do you want to go?
Yeah, can I stop? Chris, thanks for that talk. I loved it. I haven't actually seen I've heard about this work, but it's really nice to see you talk about it. I thought that when you mentioned while you were giving this talk about kind of having this remaining mixture at the end, I'm really curious about what your interpretation of posterior uncertainty on the parameter is when the model is misspecified, because you don't have you basically get a set, right? Like that's kind of the limit. As you see more and more data, it won't converge to like a point mass. And this kind of stuff doesn't really happen in like frequentist statistics, for example. You always kind of converge to the pseudo truth. But maybe this is a nice way to talk about uncertainty. Like if your model is misspecified, you shouldn't collapse to a point, right? If you see more and more data. So I'm just wondering if can you interpret the posteriors of the parameters? Like, are those kind of helpful, or are they kind of just nonsensical kind of mixtures, densities for prediction?
That's a really good question, Edwin, but I don't have a good answer. This is it's something that I think is challenging. I know that in generalized Bayes, people have thought deeply about what these generalized posteriors mean, and there's still debate going on there. And I think there's even more debate that you could have here. Pragmatically, if you want to do causal prediction in these settings, you have to use the causal frameworks that these domain experts have built, right? So that, for example, they have an ODE and they explain how you update the ODE when you've got an intervention, which terms to remove, which terms to add, and this kind of thing. And that's what you have to work with to collaborate with these people. So there's always going to have to be model parameters floating around, and they're being sort of judged on a predictive basis in this prediction-centric approach, but whether or not it makes sense to think of the prediction-centric posterior as, say, someone's uncertainty, I'm not sure it does.
I guess it's more like the parameter is sort of like, it feels a little bit closer to weights in the neural network, or they're kind of tools for modeling, right? And not so much like, oh, I want to know this parameter exactly, or want to construct intervals for it.
Oh, I see. So like deep ensembles or something, where you have an ensemble of neural networks, each with a different set of parameters, and the joint sort of collection of neural network predictions is like somehow quantifying uncertainty, something. Yeah, it has that flavor. Yeah.
Yeah, it feels a little bit like that. Thanks for that. That's really interesting. Thanks.
Jay, do you want to go?
Yeah, I actually want to kind of respond directly to what Edwin said. So, these results aren't public yet, but you can basically show that under, like, if things are well specified, these things concentrate onto the same thing that the generalized Bayes posterior would concentrate onto. So if you're happy with a generalized Bayes posterior with the MMD, at least in the limit, you actually end up in the same place if everything's well specified. And then indeed, so as you were saying, like the problem of conceptually interpreting this thing, you only get a divergence under misspecification, actually. So once again, you have this property that you kind of recover the interpretation of uncertainty in the case of a well-specified model, somewhat. And then once you go outside of that, it's kind of everybody's guess. But by the way, you were saying this doesn't happen for frequentist. Set convergence can happen in frequentist cases as well.
That's
So
true. Yeah.
if you have like a lack of identifiability, which is basically, you can think of this as like an extended model class, right? This mixture model is kind of not identifiable. So when Chris was talking about the non-parametric maximum likelihood, you kind of have the same problem there. This is why you get these weird solutions when you try to solve that problem without regularization. Yeah, anyways, sorry.
Yeah, actually, I didn't mean it as a problem. Actually, I thought it was maybe a good way to handle misspecification, like a set. Like you just tell me this is the set, and I have a measure on it, and like gives me good predictions. I think I would actually be kind of comfortable with that,
Mm.
or more than like, oh, here's my pseudo truth, which is like maybe a little bit harder to interpret in that sense. I like that as a set. Actually, I think that's really interesting.
Do we have any more questions from the audience? Yep, Sergio.
Yeah. Okay. Hi. Thanks, Chris. That was a great talk. I was wondering, I mean, are there any situations where mixtures in this parametric family are themselves misspecified? I mean, or can you show that that's not the case in this ODE example in chemistry?
Yeah, that's a good question, Sergio. Thanks. Yes, so I use this running example of if your model is Gaussian, but your data are not Gaussian, the model misspecification goes away as soon as you use a Gaussian mixture model because of the sort of universal approximation properties of Gaussian mixtures. But you're right, that definitely wouldn't completely solve the problem for more complicated scenarios. So yeah, for systems biology, no sort of ensemble of these simple ODE models is actually going to capture the true data generating distribution just because of all of the complexity going on. I mean, you know this stuff as well, right? All the interactions going on in the cell. There's all sorts of latent processes and so on. You wouldn't solve the model misspecification just using a mixture. And it would be interesting to understand how far away you are, right? So how much of the misspecification do you do you get rid of by lifting from the original model to a mixture, and how much is left? I don't know sort of a principal way of tackling that, but it's a really important issue.
Any final questions? We maybe have time for one more quick one. Otherwise, I think thank you very much, Chris, once more. Oh, for your talk, it was brilliant, and we'll see everyone again next Tuesday.
Thanks a lot, everyone.
Bye bye.